Hello everyone, it's a real honour and pleasure to be with you today, and for Patrick to have kindly set up this meeting with you all. It really is a true pleasure, and to give you a little bit of backstory on myself, I think you know a little bit of information from the email, but I'm making a film about the Aral Sea in Central Asia, and our film is focused on the different development projects that are going on to improve the ecosystem, improve the socio-economic landscape. And one of the areas in which I discovered through this project was indeed Artemia, for which they have now quite a sizable industry. This became really fascinating to me, and particularly in the last few months, I've been very dedicated to learning about this field. I'm now writing this article for National Geographic, as I think you all know. And the angle of my article is looking specifically at how Artemia can be an alternative economic resource, and already is, for communities that are grappling with climate change and water management issues. And at this moment in time, my reading and my learnings have been more focused on the Aral Sea region and GSL. So it's really, really exciting for me to be speaking with so many different people from around different areas of the world, and to hear about how Artemia is operating in all of your countries. So without further ado, I will pass the floor on.
I retired in 2013 from Ghent University as a professor of aquaculture. I got involved with Artemia research ever since 1969, but we will hear from some other person who started even earlier. First for my MSc and PhD studies with Artemia, and later, I must say, I have been working with a fantastic team of co-workers in Ghent and colleagues from all over the world in research and development aspects of Artemia. I enjoy my retirement, and I try to help and facilitate new developments with Artemia and in aquaculture. But I wish I was 30 years younger, as we live in very exciting times for more research and applications with Artemia, as we will hear in this meeting. Thank you, Saxon, for showing so much interest in Artemia to prepare an article for National Geographic. I will share the screen and I will start with my presentation. So my presentation will deal with the new initiative, the International Artemia Aquaculture Consortium. And to bring it in a perspective, we are starting back in 1960s, when only two sources in the United States were used in fact, the aquarium hobbyist. It was quickly thereafter that the Japanese were the first to see that for modern aquaculture, hatchery production, that Artemia could be used as a very ideal replacement for plankton in feeding all kinds of larval stages of fish and crustaceans. But in 1976, when FAO was organizing the first technical conference on aquaculture, it turned out that it was very risky to develop a new aquaculture system with the hatcheries when you have to depend only on sources from one single country. So this is where we try to explain that there were opportunities for other sources, other developments. And upon suggestion of the FAO in 1978, we set up the Artemia Reference Center. And most important was that right from the setup, we were looking into the possibility to bring scientists together in different disciplines to look for solutions in using Artemia. And many, many papers, manuals, handbooks, conferences were organized, which indeed resulted in improved utilization of Artemia from different sources, as we will hear in the next presentations. So quickly, we have seen already in the 1980s that Chinese, Australian, Brazilian, different new sources of Artemia came in the market. But that was also at the time that we have seen a real, I can call it an explosion of hatchery activity with different crustacean and fish species. And as you see at the bottom of the screen here, even today, we can still say that the key to successful aquaculture is the availability of hatcheries the availability of the live food sources like Artemia for these hatcheries. With such a spectacular development in hatchery aquaculture, we have seen indeed also a spectacular increase in demand, and lucky enough also offer of Artemia for feeding all these billions of fish and shrimp larvae. And the commercial sources that were developed over time, and again, we will hear in a number of presentations that the Great Salt Lake is playing an important role. Then there are a number of places in Central Asia, in China, but there are critical needs. It's very nice and fantastic to see that there has been such a development, but there are a number of issues where we need to express some concern when it comes to sustainable harvesting, when it comes to the protection of Artemia habitats, the preservation of the gene pool. In the meantime, we see that next to the sources from nature, that there have been very interesting developments in producing, in controlled production, pond production of Artemia. And in fact, in a number of countries, we will hear about in the presentations tomorrow, this is a catalyzer for local new aquaculture developments with important socioeconomic implications. Next to that, but no time to go into the details, except with also some presentations we will hear from Jim and from Wei Jun. Artemia is really a model organism in a lot of biology research. You see a number of the topics here. If needed, we can further elaborate later in the discussion. It's also nice to see that over the years, you see the red stars and you see the white stars representing Artemia research institutes, representing Artemia projects. There is a real world interest. And this came to the attention of the Food and Agriculture Organization, who really wants to see and support the establishment of such an international consortium, where a number of key issues that I addressed earlier and that are listed here without going into the details, or in fact, an opportunity to look into consolidation on one hand with Artemia, on the other hand, explore new opportunities. Thank you.
Yes, I am Jim Clegg. I've been interested in using Artemia as a model system for quite a few decades. Today I'm going to address chiefly the abilities of the encysted gastrula embryo, which you see on the slide to the left. This is a hydrated Artemia embryo at the gastrula stage, surrounded by a very tough protective shell on the outside. This embryo is capable of resisting a huge variety of environmental insults. I will give one example to start, and that is its ability to lose virtually all of its water. All of its liquid water is lost by evaporation without killing the embryo. And I've been interested in trying to understand how this embryo does something that kills the vast majority of animals of any stage. Right. Very few creatures can withstand total desiccation as Artemia can. And that's why I said some years ago that we should consider the brine shrimp Artemia as an animal extremophile, because it has this astonishing ability to withstand stresses of various kinds. To the right, you see the dried cyst. It's very difficult. We found it impossible to get electron microscope images of the dried embryos. But at the light microscope level, they are amorphous. They seem to disappear into a background, and I'll give you a reason to suspect that we know what that is. Okay, Central to Artemia's abilities of various kinds is a disaccharide called trehalose. It's a molecule consisting of two glucose molecules hooked together in a certain way. And this trehalose, we believe, does two major things that is involved with the resistance of the Artemia embryo. One is that it serves as a water substitute. By that I mean, imagine the water being lost from a cyst by evaporation, and as the water is lost, this would normally result in the damage, denaturation of macromolecules of all kinds that rely on water for stability. And that includes membranes. And of course, cells are chock full of membranes of all kinds in different organelles. Now, we believe, on the basis of some evidence, not conclusive, we believe that the trehalose replaces the water that's lost, thereby stabilizing the macromolecules and membranes. So it is a substitute in that sense. Another function of the trehalose is to form a glass at very low water contents. When the water contents get close to zero, they never really get to zero, but close, the trehalose forms a glass. And as you all know, glass is a very stable structure in the sense that we know it, but trehalose glass is also very stable, and we believe, and have reason to believe, that the components of the Artemia cells in the cyst get embedded in the glass, in the trehalose glass, thereby becoming very stable and yet becoming reversibly functional. Okay, so the two major roles of trehalose being a water substitute and the formation of a glass. Okay, so this molecule is very important. By the way, I don't think I mentioned that the amount of trehalose in an Artemia encysted embryo is massive, about 20% of the dry weight. That is a huge amount of trehalose. And so the ability of the embryo to synthesize this molecule is crucial to its subsequent stability in the presence of threatening stresses. Okay, the Artemia embryo also has the ability to undergo hypoxia, the absence of oxygen, for months and months. And it does that, we discovered, by just stopping. It just comes to a standstill metabolically, and that's quite an achievement. In that stable state, it is able to survive in the absence of oxygen for months and months. We're not sure how long, but it's certainly a long time compared with other animal forms, whatever the life cycle stage we're talking about. Okay, so desiccation and resistance to oxygen lack are just two of the very impressive abilities and capabilities of the encysted gastrula embryo. The embryo, if conditions are suitable, the embryo, and by that I mean adequate water and adequate oxygen, the embryo then develops and hatches from the cyst as a nauplius larva. The Nauplius larva then undergoes subsequent growth and molting, and eventually forms the adult male or female Artemia. One of the very impressive abilities of the Nauplii and the adult and stages in between is the ability to tolerate very high sodium chloride concentrations. Once again, conditions that are very, very dangerous for the vast majority of aquatic invertebrates. The Nauplius indeed has a very special gland in its neck called, poetically, the salt gland, which helps handle the exposure to very high sodium chloride concentrations. And so that's a unique morphological characteristic of the Nauplius larvae. The intermediate larval stages and the adult also have cells that are specialized to cope with sodium chloride. So the larvae and adults are also extremophiles in the sense that they can withstand exposure to that usually threatening condition of hypersodium chloride. So, in quick summary to the talk, Artemia really is an animal extremophile of extraordinary capabilities, and future research will continue to reveal very, very good treatments. Thank you. I could mention an application. Trehalose has been used to stabilize mammalian red blood cells to be used in battlefield circumstances, as well as storage in hospitals that lack adequate refrigeration. So that's one application of Artemia's basic science, the understanding of trehalose and how it works.
Okay, thank you very much. I'm Gonzalo Gajardo from Chile. Got involved in Artemia research since my postdoctoral days in UK, in Wales. And since then, I have been related with the Artemia Reference Center. I think I'm lucky in Chile to have two of the sexual species in very different environmental setups. In the Atacama Desert in the north, where Artemia franciscana ends its distribution, and also in southern Chile in the Patagonia, where Artemia persimilis is located. So I'm lucky about that. And I must say that I'm really intrigued about what is happening in aerial sea. So I'm grateful for this invitation. Next, Patrick, please. My ideas in this talk is that, as Jim said, Artemia is well adapted to very difficult environmental conditions that hypersaline environment impose on survival and reproduction. Number two, hypersaline lakes like aerial sea now, I read that salinity is very high now, provide multiple economic, as in aerial sea, resources like Artemia, cyst, but also in other places is mining, and also non-economic services like habitat for water birds. Number three, stability or instability of the ecosystem affect Artemia dynamics and also can influence the economic business. The number 4 is important because saline lakes are shrinking at an alarming rate. And this is also true for RLC. And one wonders how this will affect Artemia dynamics and the services provided. I see RLC as a natural colonization experiment. Artemia was introduced by wind or water birds. There is a need to monitor this experiment and the genetic characteristics of the Artemia population there, that is Artemia parthenogenetica. Next, Patrick, please. As said before, the brine shrimp Artemia is an example of adapted to critical life conditions. And as you know, because the processes of life are complicated, there is no simple solution to the problem of surviving as individuals and species. The unpredictable challenges that environments pose to populations. So in the case of Artemia, we have two solutions. Sexual species or bisexual, and asexual types like Artemia parthenogenetic. I think sexual species are really privileged during the evolution because of genetic recombination. This is important when a male and female share genetic information. Instead, asexual types like Artemia parthenogenetica, for text books, they represent evolutionary dead ends. They are supposed to be difficult to thrive in different environments. And the second point I would like to make here on the left-hand side is that human-induced perturbations have increased environmental uncertainty, greatly compromising species, ecosystem, resilience, and services like cysts being produced in RLC, and the ability to withstand such perturbations. I got this image photo from RLC from a web page. And next, please. As I mentioned, many of the world's saline lakes are shrinking at alarming rates, reducing water bird habitat and economic benefit. So this was a very important paper I read for a previous presentation. Next, Patrick. And since we are talking about National Geographic, I came across with this paper called The Rush for White Hole, and it shows how in Bolivia, Lake Uyuni has a huge deposit of lithium, so the government is planning to exploit it for economical benefit. And also on the right-hand side, we have the Atacama Desert, where salty lakes coexist with the huge, I think it's the most important lithium extraction being performed in the world. And what is the problem is that brine pumped from beneath the solar surface in Atacama Desert may impair the habitat for flamingos. Okay, next. So continuing with this evolutionary solution to survive in harsh environments, I think Artemia is peculiar, a very important example of extremophile. Well, we have 6 sexual species, as said before. Patrick wanted me to highlight some genetic characteristics, and I mentioned here the diploid number of most species is 2n42, but Artemia persimilis is one of the few with 2n44. And there are many parthenogenetic types with different levels of ploidy. And this is something very amazing. And point number 2 is very interesting also. There are 2 reproductive modes of viviparity. That means the production of diapause, cyst, and also ovoviviparity, the production of free living nuclei. Both sexual and asexual species produce both. So it's important for you to know that here are the sexual species, and I ordered them because in relation to the oldest diverger from ancestral species, Artemia persimilis, found in Chile and Argentina, as I said before, is the oldest. So you have the time passed since diversion from ancestral species is more or less 80 million years ago. So it's a good time. Then we have Artemia salina that is found in the Mediterranean area 40 million years ago. And Artemia franciscana that is very common in North, Central, and South America. And as said before, it's very important in aquaculture, 32 million years ago. Then we have Seneca, 19 or close to 20 million years ago. And then Artemia urmiana from Lake Urmia, Artemia tibetiana from the Tibetan Plateau, and several Eurasian haplotype complexes that are very young. So here on the right, you have the phylogenetic tree got by Baxevanis from Greece. So you have the similarities and differences between the species. What is very important, is shown by the red dot there. You see in black, very thick black, Artemia parthenogenetica that may be related, very related with Artemia urmiana. So when you saw the first slide and you see the proximity of Lake Urmia and RLC, you can understand why Artemia parthenogenetic could colonize RLC. Okay, next, please. This is very common, but again, just to highlight that Artemia female can produce naupli when the environment is stable. On the right, yes, thank you. And cyst, that is important for RLC when the conditions are not stable. I should say that no one knows very well what stable or unstable conditions means. Next, Patrick, please. This is just to summarize what I have said before. So you see in A minor here, what is said already. And here in B for individual, what Jim Clegg said. uh, very important metabolic and physiological mechanisms. And here on C, some very important characteristics of Artemia franciscana. There are few genetically, well, the sexual species, few genetically different species with regional endemism. Uh, in Artemia franciscana, mainly genetic diversity that is very useful for resilience is distributed in different baskets. This is like in banks with your investment. This means locally adapted population. So Artemia franciscana is well distributed in different places. So every population has particularities. I will go to cyst here that store a genetic memory of historical population condition. So this is very important, I guess, to understand also in aerial sea. And ecological adaptation demand recognitions of peer. For example, sexual species, they need to, I wouldn't say courtship with mates, but they do this. Next, Patrick. Just to summarize, you have on the left Artemia franciscana, okay, New World species, very widely distributed. Of all Artemia species are very well studied. Equipped to survive in many environments due to the high genetic variability, genomic and transcripts variability, genes involved in cyst formation, salinity, and temperature tolerance, hypoxia, as mentioned, are upregulated and downregulated depending on the conditions. Cysts can be viewed as gene banks that store a genetic memory of historical population condition. In sexual species, female can mate males. This is weird, but it's true, from the past, from the present, or from the future. And there are some studies that show that female prefer contemporary males. This is amazing. How do they do it? Is interesting. Well, I go to Artemia parthenogenetica, not to take much time. It has a restrict distribution. Recent origins, I've mentioned it before, multiple origin events in Central Asia from Artemia urmiana and probably from all the sexual species. Less variability due to the lack of genetic recombination, and because of that, they thrive in unusual habitats. That means different from those occupied by parental sexual species. And what is surprising, and I will mention this in my last slide, parthenogenetic forms come displace, can you go back? Can displace sexuals under very special conditions. I wanted to finish my presentation saying that aerial sea is a very nice natural experiment that needs monitoring, as we have seen here in Great Salt Lake. Particularly the genetic characterization of the existing Artemia. I think I couldn't find more information about Artemia parthenogenetica that is in the sea. And also, I think if Artemia parthenogenetica originated from sexual Artemia, the upcoming Artemia Franciscana genome will provide information on how much Artemia parthenogenetica owes to sexual Artemia. So next, please. This is the paper that is soon to come out, where Jim and Patrick and a lot of people from Belgium are involved. So the genome will facilitate research on most extremophile features, providing reference sequences for crustacean research. And my personal opinion is that it will help in understanding many of the features that have made possible that Artemia parthenogenetica evolved in Aerial Sea. Okay, thank you.
My name is Marcos Camara, and it's nice to be here today with you. I am a full professor at the Federal University of Rio Grande do Norte in northeastern Brazil. And during my talk, I will summarize the development of Artemia production in the coastal saltworks of Rio Grande do Norte state. I will also review current cyst harvesting practices and try to outline why these harvesting practices impact future cyst yields. And finally, I will indicate a science-based approach to securing the productivity and persistence of this biological resource. Well, as you have mentioned before today, it all started in Kyoto in 1976, when Artemia was considered problem to the expansion of aquaculture. And Patrick argued that this shortage was an artificial and temporary problem, and he undertook efforts in the following years to make this production of Artemia viable in developing countries. As Eddy Naessens just mentioned, and the establishment of ARC at Ghent University upon suggestion of FAO in 1978, and the organization of several international Artemia training courses thereafter. And by chance, the first international Artemia training course was in Natal, my hometown, in 1977. And here, Patrick. And it made viable the first inoculation of Artemia in Brazil, in Macau Solar Salt Works. And before going into more detail, I'd like to make this connection, because aquaculture in northeastern Brazil is practically synonymous with shrimp farming and Artemia. These two systems are conceptually different, but they are closely related. Shrimp culture systems employ industry standard technologies, but Artemia are produced in more limited quantities and through the use of artisanal methods, as we call it, extractive aquaculture. Okay, in this table, you have an idea of production figures for Lithopneus zonamei, which is the global shrimp species, and it's also farmed in Brazil. So in 2020, Last year, production was around 90,000 tons, no, 30,000 hectares of production area. So 20 billion shrimp pulse larvae were produced. And this amounts to about 20 tons of consumption of Artemia systems, around 4 tons. So around 20% of these demands are fulfilled with cysts harvested in local salt fields. So you have some pictures here. So the production of Artemia cysts are quite relevant, and it's a form of extractive aquaculture in Rio Grande do Norte. These cysts are harvested in salt works as a byproduct of the marine salt industry, which produces about 95% of all marine salt consumed in Brazil. So it's quite important to the economy of Rio Grande do Norte state. The main cyst producing areas are located in and around the municipalities of Macau and Areia Branca. Also here in Grossos, around Mossoró. This is the capital, Natal, where the main campus of Federal University of Rio Grande do Norte is located. And this is where I live. So a few details about also the use of juvenile and adult Artemia biomass. So it's also collected in these huge salt production operations. They are harvested, frozen, freeze-dried, or made into flake diets for larval, post-larval shrimp. This is quite important because although the quantities and price of these biomass represent just a small fraction of those of cysts, this activity represents an additional and more sustained income to fishermen. Well, I will just briefly talk about farming Artemia in northeastern Brazil. So it was technically successful, but operational costs, basically inorganic fertilizers and labor, inhibited the consolidation of this initiative. So cyst production still remains as the only form of extractive aquaculture in Rio Grande do Norte. In these pictures, you can view some of the different steps, scooping the cysts from the water or from the shore, cleaning, storing in brine, sun drying, and cysts ready for sales. Well, and to finalize my presentation, I'd like to show you some of the challenges I had to a sustainable use of Artemia in northeastern Brazil. So for many years, cyst harvesting has been an important source of income for small-scale fishermen and their families. So it's imperative for us to realize the importance of these small-scale fisheries to aquaculture and to rural livelihood development. And on the other hand, it's also important that we mention that over-harvesting of cysts has been exacerbated because access to this fishery is open and unregulated, differently from what we have heard before here regarding Great Salt Lake, for example. So, and because of this scenario, simple artisanal technology gives fishers the ability to overfish. So quite a big challenge, try to regulate this harvesting, because there are indications that overharvesting of cysts has caused a directional reproductive shift to ovoviviparity, direct production of free living nuclei in local population of Artemia. So there is an urgent need to clarify whether harvesting is inducing genetic changes and if plastic and evolved responses are leading to lower and, of course, unsustainable cyst yields. Well, some of the thoughts I have presented here are detailed in this publication from last year after the gold rush, review of Artemia cyst production in northeastern Brazil. And I finally like to acknowledge the contribution of these institutions, okay, by alphabetic order, Artemia Reference Center, Brazilian institutes, Canadian agency, European Commission, FAO, IFS, and of course my university, Federal University of Rio Grande do Norte. And finally, also, I'd like to dedicate this brief presentation to the hundreds of small-scale artisanal fishermen from the salterns of Rio Grande do Norte. Over the past 4 decades, these cyst prospectors have made brine shrimp production in northeastern Brazil a reality. Thank you.
My name is Don Leonard. I'm currently the chair of the Great Salt Lake Brine Shrimp Cooperative and have served as chair for the past 15 years, and also as CEO of the cooperative for 14 of those 15 years. I've also served as president of the Utah Artemia Association for 27 years. I've been asked to take a few minutes to share the history of commercial harvesting of Artemia from Great Salt Lake. Let me first share a few details about Great Salt Lake. Great Salt Lake is the largest saltwater lake in the entire Western Hemisphere. It's the 4th largest terminal lake in the world, which is why it retains a level of salinity that's ideal for Artemia. Great Salt Lake's the 37th largest lake in the world. Its dimensions are 75 miles by 40 miles, or 120 by 64 kilometers. And it does fluctuate in size due to annual variations in precipitation, but its average size is 1,730 square miles, or 4,352 square kilometers. It's important to note that Great Salt Lake is a Western Hemispheric shorebird reserve. Millions of birds rely upon Great Salt Lake as a stopover to gain mass and strength as they migrate. The current salinity of Great Salt Lake is 14%, or about 140 ppt, in the south arm, where the Artemia flourish. But in the north arm, where it's too salty to sustain independent populations of Artemia, the salinity is 26%. The Artemia industry on Great Salt Lake started in 1949. That's 72 years ago, when a tropical fish enthusiast from Ogden, Utah, named Cleon Sanders, heard that adult brine shrimp were a nutritious food that he could feed to his tropical fish. Initially, he had purchased frozen adult brine shrimp from San Francisco Bay. Then he heard from a friend that Great Salt Lake, right here in his own backyard, had large quantities of the tiny brine shrimp. A year later, in 1950, Sanders authored and sent an article to the Aquarium magazine describing his fish food from Great Salt Lake. Shortly after his article was published, he began receiving numerous requests for frozen adult brine shrimp from around the USA and formed Sanders Brine Shrimp Company to respond to those requests. In the 1970s and 1980s, the farming of various species of shrimp and fish was expanding globally. Newly hatched Artemia nuclei had been found to be an ideal food for larval stage shrimp and for some species of larval fish. With the growth of aquaculture, the focus of the Great Salt Lake brine shrimp industry switched from harvesting adult Artemia for use by fish hobbyists to harvesting Artemia cysts, also commonly referred to as brine shrimp eggs. It's important to note that, as Jim stated, cysts can be processed or dried and packaged for long-term storage, so they can be shipped around the world while maintaining their hatchability, their nutritional quality, and their other valuable characteristics. With this rapid growth of aquaculture, demand for Artemia cysts grew quickly, and by the 1990s, the Great Salt Lake brine shrimp industry had grown from 1 to 32 harvest companies. At that time, this increasing harvest pressure on the lake caused many of these companies to become concerned about the long-term health of the brine shrimp population and the resource. You have to remember that by this time, in the early 1990s, excess harvest pressure had already damaged or destroyed numerous fisheries around the globe. For example, the Peruvian coastal anchovy fisheries crashed in the 1970s. The sole fisheries in the Irish Sea, the West English Channel, and other locations had become overfished to the point of virtual collapse. Blue walleye went extinct in the Great Lakes in the 1980s. But it was the collapse of the cod fishery off the coast of Newfoundland in 1992 that really caught our attention. In the summer of 1992, the northern cod biomass fell drastically to just 1% of earlier levels, and Canada declared a moratorium on cod fishing. Of equal alarm, biomass available to feed the cod, or the cod prey species and food chain, virtually disappeared, undermining the ecosystem and leaving little hope that the cod could recover. In addition to the collapse of other fisheries around the world and to the rapid increase in harvest pressure on Great Salt Lake, we were concerned about overfishing from Great Salt Lake because there was no formal state program to manage the brine shrimp resource. So there were no limits on harvest permits, nor limits on quantities of product that could be harvested. There was no significant state research program for the brine shrimp resource, so there's no understanding of how to preserve and protect Great Salt Lake Artemia. And there was no state mandate to regulate the brine shrimp resource. So the Great Salt Lake industry approached the director of the Utah Division of Wildlife Resources and asked that the state establish a program to manage and protect the Great Salt Lake brine shrimp resource. In support, the industry offered to provide additional ongoing funding for research, management, and compliance. With this new research data and with the support of the industry, the Utah DWR established a sustainable management model for brine shrimp in the Great Salt Lake. Since all adult Artemia in Great Salt Lake die in the winter due to cold, lack of feed, or other causes, the Utah Division of Wildlife Resources determined the number of brine shrimp eggs that must be left in the lake at the end of the harvest season, which ends on January 31 of each year, to assure that the brine shrimp population would reemerge each spring. That equation, developed by the Division of Wildlife Resources, takes into account such factors as winter cyst survival, avian requirements, egg hatchability, and other factors. This sustainable management model allows the industry to harvest only the excess Artemia cysts, with that excess being determined to be those cysts above 21 cysts per liter of water in the Great Salt Lake. This resource management model has succeeded beyond everyone's expectations. The result is a natural, sustainable brine shrimp resource that supplies both the avian populations and the industry with vital Artemia cysts, while preserving a healthy, natural population of brine shrimp in the lake. The state of Utah's resource management plan for Artemia has been so successful that it is touted as a model of successful government and industry collaboration. As a result, Great Salt Lake is a reliable, in fact, it's the most reliable Artemia resource in the world. It's the world's largest supplier of quality Artemia cysts. And of important note, annual average supplies of Artemia cysts from Great Salt Lake have steadily increased since implementation of the state's management plan. And GSL is by far the world's most sustainable Artemia resource. The state of Utah's Artemia resource management plan will continue to preserve and protect the Great Salt Lake brine shrimp resource because Utah established the Great Salt Lake Ecosystem Program to regulate the harvest, to research and monitor the resource, and to preserve and protect a healthy brine shrimp population in the Great Salt Lake. At our request, The state also established a standing technical advisory group, which is a science peer review committee to oversee research on brine shrimp in Great Salt Lake. The industry contributes funding for that research. We also share our own ongoing research with the state, and we serve on the state's technical advisory group. So we're very optimistic about both the quality and the supply of Great Salt Lake Artemia going forward. Thank you.
Hello, good morning, good afternoon, and good evening, everyone. So first of all, I should thanks a lot for the invitation from Saxon and Patrick. My name is Liying. I'm the professor at Tianjin University of Science and Technology, and I'm also the director of Asian Regional Artemia Reference Center. My experience on Artemia research and application can be dated back 30 years ago. In the meantime, I have obtained my master and PhD doctor degree at Ghent University under the supervision of Professor Patrick Sorgeloos. Next, please. Patrick, please. Yes, so I would like to start my presentation with the history of Artemia resource exploration and utilization in China. China is rich in Artemia biodiversity and resources. Artemia sinica and Artemia tibetiana are the two indigenous Artemia species, while parthenogenetic populations also exist in the salt works and the salt lakes. Since early 1990s, Artemia franciscana have been existed in the Bohai Bay salt works as the result of inoculation of Artemia cysts from San Francisco Bay. Next, please. Before 1980s, the Artemia cysts and biomass are usually used as a supplemental feed for the local chicken farm, resulting in a bright orange color of the egg yolks. However, the commercialization of Artemia resources in China only started from later 1980s, along with the fast development of marine shrimp and Chinese mitten crab larviculture. Nevertheless, the yield and quality of Artemia products are unstable and unsatisfied. The study was initiated in 1985 at Salt Research Institute of China about Artemia, with the support of the UNDP of the United Nations and the EEC, the previous EU. The overseas Artemia expertise, especially the team from Ghent University, have made very important contributions to the initiation of sustainable exploration and utilization of Artemia resources in China. In the following 10 years, many progresses have been achieved. Next. By working together, people have made long-lasting friendship. So you can see the face of young Patrick more than 30 years ago. Next. China has the world's largest market demand on Artemia cysts. It is also the #1 cyst producers and importers in the world. China also exports products to the other Asian countries. Next. At present, more than 70% of the cysts are sold to the shrimp hatcheries. Except for the cyst products, Artemia are also sold in form of newly hatched Artemia nuclei, fresh and frozen biomass, as well as decapsulated cysts, to be used in shrimp hatchery, nursery, and broodstock maturation. Next. Owing to the vital role of China in Artemia resources producing and consuming, and to the fast development of aquaculture industry and increasing Artemia consumption in Asia, FAO expressed their interest in establishing a regional Artemia reference center in Asia. In November 2016 FAO experts workshop was successfully organized in Tianjin, China. Next. The establishment of Asian Regional Artemia Reference Center in China was recommended by the participants at the workshop. With the tremendous support of FAO and our university, Tianjin University of Science and Technology, the laboratory set up at ARARC has completely in the time of 1 year. So in April 2018, next please, the official opening of ARARC and Artemia workshop were held in Tianjin. Next. So the role of ARARC is, of course, to facilitate the sustainable exploration and utilization of Artemia resources in Asia through coordinating interdisciplinary and interregional Artemia research, setting up platform for Artemia information exchange, providing technical consultation and services for Artemia cyst production and its application in larviculture. Next. We have quite young staffs with specialized experiences and innovative consortiums. Next. And we have also advanced the laboratories and research facilities at ARARC. Next. ARARC have preserved more than 300 Artemia cyst samples and set up the database and providing and exchanging the samples with the other institutions for scientific research purpose. Next. Since 2019, we took initiative to survey the Artemia resources in the salt lakes in Tibet. The world has the plateau. Tibet Artemia has unique phenotypic characteristics in terms of dark red color, the very big size of cysts and high EPA and DHA content. Lots of Artemia samples were collected from the altitude above 4,000 and 4,000 to 4,500 meters. Next. The services to the aquaculture industry is one of the main tasks of ARARC. We provide the technical supports and training courses to the private sectors, to the communities in China and in the Asian region. Next, please. ARARC has made efforts for the science popularization. We have set up an Artemia laboratory with, at the National Maritime Museum of China and provide short courses to the visitors. Each year, ARARC have also received lots of students from the local schools to learn about Artemia and the other marine organisms. Next. Our PhD, our master and undergraduate students have also contributed a lot to the Artemia researches and the practical work at ARARC. Okay, next. The last but not the least, I would like to share our recent fundamental research results related to Artemia biology. As you all know, Artemia is such a unique zooplankton with special life phenomenon. For example, the high salinity tolerance, the ovoviviparous or oviparous or ovoviviparous offspring production, the dormancy and diapause termination, etc. So it is necessary, we think, to make Artemia as a model organism for scientific research. Patrick, please click one by one. Yes. So far, the cerebral garland morphology and structure, the classification and the cytological characterization of the hemolymphy, the ovarian development and oogenesis mechanism, and the morphology and the cytological characterization of Artemia gut have been studied at ARARC. Most of these results are the brand new and, or I can say the original study. We believe that this will facilitate the progress of developing Artemia to model organisms in the near future. That's all for my presentation. Thank you very much.
Hello again. Thank you so much for that brilliant presentation. Probably award for the most beautiful backdrop of this meeting as well, with what you've got behind you. And also brilliant to see a young Patrick there looking like a 70s rock star. Thank you so much for that. Many questions coming to my mind, but I believe we will pass the floor on now to Nguyen, please. Thank you.
Hoa, you have the floor. You switch on camera and unmute, and you can start.
All right. Hello, everyone. To continue with the, can you hear me, Patrick?
Yeah, sounds great.
Okay.
Okay, that's fine. Because it seems to be that the connection from my side is not very smooth, so I try to do my best. To continue with my presentation, first of all, most of you know who I am. My full name is Nguyen Van Hoa. I'm now a lecturer in Consortium University. So we have a long-term collaboration with Ghent University for the Artemia research and application in the Mekong Delta, Vietnam. So my presentation today is to bring up only the socio-economic aspect of Artemia farming and the solar source work in the Mekong Delta. Next, Patrick. So for some of you, you know already where Vietnam, but the rest just consider that Vietnam is located in the Southeast Asia. And Vĩnh Châu location where we're going to do the Artemia stay in very south of the country, where you can see that the Sóc Trăng and Bạc Liêu and the SC cultural line. Next, please. So the figure here to illustrate that the activity on Artemia culture or production in Vĩnh Châu and Bạc Liêu since 1986 until recently. So to bring up the information about the culture area, the total production and the yield, they are go up and down because of so many factors. But up to now, the area they can produce up to 60 tons of the wet weight of Artemia cysts, and they can fulfill the need of the local aquaculture perfectly. So if you follow the course of the time, you see that the different culture system or farming system have been applied. First, we start with traditional, and then it follow with intensive system, and now also intensive, but we apply the biofloc technology with the aim to save the environment and also to enhance the seed production. Next, please. So this graph to bring up the figure that where you can see that we started in the year of 90, a little bit earlier of 90. But up to 90, you see almost 2 hectares of Artemia farming with 100 families involved. And later on, up to the year 2000, the culture area go up to 1,000, where you have 500 families. And 2015, already it increased up to 1,200 hectares, 1,200, and more than 1,000 families. So it means that 3, 4, 5 farmers involved. However, until the year of 2020, only 800 hectares from 500 family involved because of different region, where you can see that most of them, they also refer to do for the shrimp farming or the local area, they switch into the wind farm project for energy. At the meantime, the big problem that the local people they have to face with is the abnormal weather, where you can see that the early rainy season and also very short dry season. Therefore, the production area become less and less. However, we try to enhance the Artemia production by improved technical protocol. Next, please. So to bring up just the information how Artemia culture system look like. So if you have time or if you have the chance to visit the site in Vĩnh Châu and Bạc Liêu, then you will see the source street belong to the family or the household. Normally, they may have the area up to 1 or 2 hectares, or sometimes 5 to 10 hectares, it depends. And where they can split up into different ponds. And each Artemia pond, they have the area up to 0.3 to 0.5 hectares each. And we keep the shape of the pond a rectangular shape. And over here, we also try to design the diagonal coincided to the revealing wing, the local wing direction, in order to facilitate for the seed collecting. And to avoid the heat, the high temperature during the dry season, especially to the end of the dry season, then we try to elevate the water level up to 40 or 50 centimeters from the platform. And of course, the system needs to be easy to accept with the seawater or saline water in order to maintain the salinity throughout the culture period. And don't forget that we have to keep the fertilization pond up to 20% or 30%. It depends on how good the local or the wild algae to develop in the area. Next, please. So for the culture protocol, I'm not going to go into detail, but you can consider that when you started with the rice cyst, after you put into the normal seawater, then it's going to have after 24 hours. From then, you can stop or you can inoculate into the Artemia pond. So weekly they will develop to become adult within maybe a week or 10 days. So they start to have the mating couple, and a few more days they're going to release either cyst or nuclei, because Artemia they could release both of them OV period or OVV period. So where the people can collect either cyst or nuclei in their farm. Okay, go to the next, Patrick. The red slide, you see how the farmer going to collect the cyst. But this slide to indicate that they can collect the alive biomass in the pond. Until now, we have the culture technique or culture protocol. They can go up to the production of 1 ton of biomass per hectare per month. It means that during the dry season where the saline water available, so then we can produce up to 5 or 6 tons of alive Artemia biomass. So this one we have different kind, different form of biomass, where from the alive biomass you collect them to put in the harponet, and then later on you can put in different packets, like chilic biomass or alive biomass or frozen biomass. It depends on the demand in the local market. Next, Patrick. So if you look at the traditional activity of the source farmer in the site, you can compare how much that for those who switch into Artemia production they can get. So the activity of Artemia in the site have been lasted under the almost 30, 40 years. And they indicated clearly that the income from Artemia farming, they can go a high, a 3 up to 5-fold compared to the source production. Then the total income in Artemia production per 6 months for the traditional model, it means that the first system, the first Artemia farming have been applied in the site, they can make the income up to $3,500 or $4,000 per hectare. While at the meantime, in the recent time, the farmer who go for intensive system, they can go up to $7,000 or $10,000 per hectare. So this is just the figure in the field. Then you can see how the people are busy with their activity. Netly. So we have started with the Artemia production, and on your left-hand side with the tin can from Consortium University. But nowadays, the activity has been expanded into different enterprise, like Vĩnh Châu Artemia Cooperative, Bạc Liêu Artemia Cooperative, Vinga Company, and Kim Sung Company. Now, every year, the whole area of up to 1,000, 1,200 hectares, they can produce up to 20 tons of rice seed Artemia with approximately $4 million of US dollar per year. This is just bring up one example of how to apply the Artemia produced in Vĩnh Châu and Bạc Liêu to the targeted species in the Mekong Delta. And this is the mud crab, Sinla paramagnoson. You know that most of the time before if the farmer want to grow up the mud crab, they have to go to the field to collect the wild crabling. But since we have the Artemia produced in the site, we can feed them directly. If you go back into the mud crab hatchery, normally that in the first stage, I mean from the jar 1 up to jar 5, normally that you have to feed them with rotifer. And for the rotifer, that is very good live food for most of the crustacean, like shrimp, like mud crab. However, to produce rotifer, it's very complicated and easy to get contaminated with pathogen. Why? One of the invention, we are a bit proud about that when we feed the mud crab larvae with Artemia in the umbrella state, where you can see the size almost similar to rotifer. Even they can be fed from jar 1 and jar 2 just after the hatching. Then the mud crab larvae, they can continue to grow, and up to now the activity can be expanded into different backyard hatchery tend to the umbrella state of Artemia. And nowadays there are more than 500 mud crab backyard hatchery. Possible in the year 2016 we even have more, and they could produce up to from 1 to 10 million of crablet per hatchery per year, and the whole area they can go sometimes up to 1.6 or 2 billion of crab annually, and it can certify to all the farmers for their rollout system. So here you are the photo on the slide to see that different culture system. They can go for intensive or intensive. If you are interested with information from the status of Artemia culture in the Mekong Delta, Vietnam, please go through the article have been published in the World Aquaculture just last year. And another paper I have mentioned about Artemia omelette, where they can make also excellent food for the human being. So if you have the chance on the Thai to visit the Vinh station, I'm going to cook that one for you, and you are all welcome. Next, please. So that's all, Patrick. Thank you very much.
Brilliant. Thank you so much for that very, very informative presentation. More information about the Artemia omelette, something that I really must try in the coming months. So I think we're going to give the floor now to Montakan.
Sorry, it seems that Wei Jun is not seeing the shared screen.
Okay. Hi, now it's okay, Gilbert. It's okay.
You're right.
Thank you.
Okay, good morning, good afternoon, good evening, everybody. I'm Montakan Tamtin, a fishery biologist from Department of Fisheries. I'm now the director of Samut Sakhon Coastal Aquaculture Research and Development Centers. My research area is focused in aquaculture management and nutrition in economical important species. Actually, after graduate, I'm right out from my university in 1992. I started working as research assistant in Artemia project under the cooperative between Thai and Belgium government. And in 1997, I also obtained my master's degree from Ghent University, and Professor Patrick Sorgeloos is my advisor. And let me start about, next please. Let me start about the history of Artemia production in Thailand. Actually, it started in 1977 when Department of Fisheries had got a microbiome project from FAO under the UNDP. And the microbiome, the development at that time, they want to develop microbiome culture. And so they initiate the Microbiome Development Center at Chakheng Sao province, which is in the eastern part of Thailand. At that time, the Artemia cyst is expensive, but it has a very high potential as a life feed for microbiome larvae. So the OF attempt to develop and culture to produce the Artemia cyst. And in 1988, there were the Department of Fishery officers participate the Artemia training course at ARC in Ghent University. Yes, next slide, please. And in 1978, there was the first attempt of the Department of Fisheries to produce Artemia cyst, which was achieved successfully in 1979. At that time, they inoculate San Francisco Bay strain and is produced around it produced 10 kilograms of dry Artemia cyst in 0.24 hectare area of earthen pond within 45 days. Next, please. And at that time in 1979, so Thailand became the first commercial producers of Artemia cyst in commercial South farms, following the successful demonstration of seasonal Artemia production in South farms in the Philippines. And later in 1980, Artemia farming for cyst production was launched by the Department of Fisheries in the South farms. And at the time, it's just in Chonburi and Chakheng Sao and Samut Songkhram. For the first two provinces, is located in the eastern part of Thailand, and for the Samut Songkhram is down to the south from Bangkok around 1 hour. And later on, there is spread to the other province of Thailand, like in Phetchaburi and Chanthaburi and also Pattani. Next slide, please. In the past, they can produce 600 kilograms of raw Artemia cyst from 5.7 hectare in earthen pond. Next slide, please. Next slide, please. Yes, in order to stimulate and enhance the application of Artemia in shrimp farming and fish farming, there was the Artemia project initiated in 1992 and 1996. under the cooperation between Thai and Belgian government. And this project is the extension from the BADC and DTEC cooperation project, which was started earlier. And that project was ended in 1978. At that time, there was the establishment of the National Artemia Reference Center at the GSL University. Next slide, please. Currently, the cyst production in Thailand has been replaced by biomass production because they can produce quite low amount of cysts, and also the demand of the farmer, they prefer to obtain biomass production of Artemia. And moreover, the high saline water from the Artemia pond, there has the high demand for saline water to use in the hatchery. And although there is quite few farms in Thailand, around 20 farms, but there is the Banjong farm, which is the one of among the farm that they produce and have the activity about the Artemia production for more than 20 years. And now the Banjong farm produce the Artemia biomass for selling, as for together like a selling, and also use within his farm. For selling, he sell to the ornamental fish market, and also for the hatchery of both Montakan and Panama, and both in the live and also frozen form. Next slide, please. And this is I show you the Artemia production from farm of Mr. Thanan. He is the alumni of Ghent University. After he resigned from Department of Fisheries, he doing his Artemia production business and farming. And nowadays from Thailand, they have the potential to produce the Artemia biomass around 400 kilograms per hectare per day, and can produce 500 and 600 tons annually. Around 75% used for local fish and shrimp, and 25% for export as a frozen product. And then this slide showing you the frozen form of Artemia biomass from Mr. Thanan, and also he produced in a live form. Next slide, please. Okay, and this final slide is almost, I think, 40 years. And Mr. Anand is the first government officer that participated the training course in 1978. And also Professor Patrick Sorgeloos, and they met each other again at the Royal Sea Farm project, which has the development of new technique of harvest of Artemia biomass. Thank you very much.
Thanks to Patrick and others for being here in this meeting. I am Naser Agh, professor of aquaculture from Urmia University, Artemia and Aquaculture Research Institute. I did my bachelor and my master in India, and I did my PhD in Artemia Reference Center, University of Ghent, under the supervision of Professor Patrick Sorgeloos. I've been working in Urmia University since 29 years, and for 26 years I've been working with Artemia. I founded Artemia and Aquaculture Research Institute, and I'm director of the institute, and also I founded the Iranian Aquaculture Society, and currently I'm president of the society. I did many national and international projects on Artemia, and currently working on Artemia production almost in many provinces in Iran, and also supporting Kazakhstan, Uzbekistan, and Russia in some sort with some private companies. So why we started working on Artemia here in Urmia? Because the Lake Urmia is the second largest natural biotope of Artemia in the world, it was 5,500 square kilometer. And it is located just 25 kilometers away from Urmia city and Urmia University, where we are working. It was first report and presence of Artemia in this lake was done about in 1900 by Gunter, and in 1976 Clark and Bowen called it Artemia urmiana, and later me and my colleagues we reported presence of parthenogenetic Artemia in the lagoons, and also coexistence of bisexual and parthenogenetic Artemia in the Lake Urmia itself. But the idea of commercial exploitation of Artemia from Lake Urmia started from the time when the first resource assessment of Artemia in Lake Urmia was done by ARC, led by Professor Patrick Sorgeloos, from 1991 to 1995, when they reported presence of very huge amount of Artemia in the Lake Urmia. After that, the Iranian Fishery Organization tried their best to convince the environmental protection authorities to exploit the lake, but unfortunately they did not give permission. Later, I started my own efforts, and finally they agreed in late 1999, 101 tons of cysts, and in year 2000, 103 tons of cyst was collected from Lake Urmia. But due to the desiccation that happened, and turned into a big crisis in the Lake Urmia, the harvesting was stopped since then. And therefore, we started, pond culture of Artemia since that time. The first attempt that I did for the pond culture was in Khuzestan area in south of Iran in 1998. Where we just, we had only a few small ponds just to show or prove that Artemia culture could be possible using Persian Gulf water. The salinity, of course, was increased by evaporation, and it was really successful, and which was a pilot project for further activities in large scale. The second attempt, it was in between 2000 and 2002 in East Azerbaijan, just near the Urmia Lake. At that time, we could collect the water from the lake itself by pumping into the ponds. We had 12 ponds, and we started producing Artemia urmiana and parthenogenetic Artemia. And the algae was used as the food source. We could produce 25 kilograms of dry cyst per hectare per month, and 2 tons of live biomass per hectare per month in intensive culture in this area. The other attempt was in Kerman province. Kerman province is in the central area of Iran. It is a very deserted area near Rafsanjan city. Of course, the first attempt there was done in 1986 by another group, but it was stopped in just 2 years. And we restarted this project in 2015. I brought the cyst from Vietnam. I collected that from Dr. Hoa. It was Artemia franciscana VC strain. We had one large 35-hectare large pond. Of course, currently another 100 hectares is under construction in the same area. There we use the salty groundwater because it is central area, deserted area, and there is no any connection to the seawater. So groundwater are quite salty, and we could use that. Of course, that area is also very hot, so salinity increases by evaporation. So in many areas, we use this kind of unconventional water sources, which you can see in coming slides also. So you can see this large pond. It was just like a small lake of 35 hectares, where we use the grinded fermented wheat bran as food source. Of course, chicken manure and molasses are also added to facilitate biofloc production as a food. And the produced Artemia in this area is commercialized within Iran. It is Artemia, of course, I said Artemia franciscana, and annually 2 tons of wet cyst and 15 tons of biomass, live and frozen, which are sold to mostly to the fishery, to the hatchery, shrimp hatcheries, and also to the aquarium pets. In another attempt in Bosworth province in 2013, I did an integrated culture of algae, artemia, and shrimp in the ponds at the vicinity of to each other. Culture water is pumped from Persian Gulf. Salinity is increased by evaporation. There also we had fertilizer ponds just to improve the algae. The natural algae is available in the Persian Gulf, so they bloom very fast. And there we produce only artemia biomass to use directly as a live food for the shrimp culture in at the vicinity ponds. This was also a successful attempt, but currently the Iranian Veterinary Organization does not allow anymore to do artemia besides the shrimp ponds, which is very unfortunate. Currently it is almost completed it was under construction since last few years. It is almost completed now. It is in East Azerbaijan, other side of the Lake Urmia, near Tabriz city. There we constructed a 55-hectare large pond, which is separated from the Lake Urmia itself. Here at the right, you can see this is the Lake Urmia, which is dried. Of course, the Lake Urmia dried by about 50% during last 25 years. So we separated a part of the lake, 55 hectares of the lake by a dike, by earthen dike. And here also we are using another unconventional water. We are using effluent from the urban treatment plant as a culture medium. Of course, the salt is available, plenty of salt is available here because after drying, the Lake Urmia left about billions of tons of salt on the floor of the lake, which is available to increase the salinity to any level that we wish. So you can see for this purpose, we had to transfer this treated water by a 10-kilometer long, 50-centimeter diameter pipe. It was a really big project. Initially, the water is coming into this big concrete pond for the pumping station, and from here it is pumped through this canal, through this pipe of 10 kilometers, and brought to this large lake, which of course the production will start from next season. The main goal here is production of at least 4 tons of dry Artemia urmiana cyst and 30 tons of biomass annually. And another project which is now currently going on, and it is again in West Azerbaijan, in other side of the Urmia Lake, near about 2 hours from Urmia city. There we have 2,000 hectares of earthen pond site, which was constructed for the fish culture about 25 years ago, but due to lacking of the fresh water, it was left unused. So we proposed to convert it into Artemia culture ponds, and 200 hectares will be in the first phase, and currently 100 hectares is active. Production is Artemia urmiana and parthenogenetic Artemia. And here again, we are using salty groundwater. It's surprising to know that in this area, which of course is not far away from the Lake Urmia, of course, the dried part of the Lake Urmia, the salinity of the ground water is about more than 100 grams per liter. So it is very ideal for the Artemia production. So micronized wheat bran and molasses are used as food source and also to stimulate biofloc production. This is a few more pictures of the currently active ponds in this area and the water which is pumped from the well. So this water, as I said, it is more than 100 grams per liter salt water. In this site at present, mostly they have focused on the biomass production because it has a good market. So they produce about 20 tons of biomass, and it is marketed also in this area and also in the southern part of Iran to the hatcheries which are fed to the broodstock mostly. And more new sites are now in Iran under construction, and 200 hectares in Bosworth province in south of Iran for Artemia, specifically for Artemia franciscana culture. Again, Persian Gulf will be used as culture medium. And another in Sistan and Balochistan province, which again in the in the southeast of Iran, near the Chabahar city, and again for Artemia franciscana culture. And in this place, we are using another unconventional water that is salty effluent from desalination plant. And also, of course, water is pumped from Oman Sea in order to regulate the the water salinity. So we are using both water to combination of both water. And of course, this kind of desalination plants are increasing in southern parts of Iran. And we had a meeting with another group, which recently integrated a big project producing daily 200,000 of cubic meters of fresh water. And they are increasing it to 1.5 million cubic meters of fresh water every day. And they have same amount of salt water with about 70 grams per liter salinity, which is again ideal to start Artemia culture, especially for the biomass production, which of course salinity increases there very fast because it is very hot area. And we propose them to construct very large artificial wetlands of at least 500 hectares each, which can be used as also for Artemia production, and also as artificial wetlands for the birds, and also for the people to just go there to enjoy. And this is our Artemia and Aquaculture Research Institute, and it's surprising to know that I started with just a 25 square meter Artemia research lab with the guidance of Professor Patrick Sorgeloos from the same year that I started knowing Professor Sorgeloos, and currently it grew to more than 9,000 square meter of Artemia and Aquaculture Research Institute. And of course, we also established a regional Artemia processing reference center. And also we have started, of course, again, this establishment of Western Asian Artemia Reference Center, which will be supported by FAO. It started about 15 years ago. Professor Sorgeloos visited, and also later Professor Gilbert Van Stappen visited, and they produced a document to FAO. And now we are again started to reestablish this indeed, and we will soon we will have this regional Artemia reference center to be established here in Oromia. That's all. Thank you very much.
So thank you. Good morning from Nairobi, Kenya. My name is Betty Nyonje. I did my MSc in aquaculture in Ghent University at the Artemia Reference Center. And I've worked with the Kenya Marine and Fisheries Research Institute all my working life. It's only about 5 years ago that I was seconded to the ministry. And ever since that time, I've been working in the Secretariat for the Blue Economy in the Executive Office of the President. And I also dabble as a technical advisor on the high-level panel for sustainable ocean economy in the Executive Office of the President. So I'll give you a brief overview of the history of Artemia farming in Kenya. Now, our greatest partner in this journey has been the Artemia Reference Center in Ghent University. Next, please. Next slide. So the map you see there shows the salt belt of Kenya. It is, if you see here, it's an area in the north coast of Kenya in a division called Magarini in Kilifi County. So the salt belt comprises 10,000 hectares of salt works, consisting of 7 corporate farms and several artisanal salt farms. Now, Artemia franciscana was introduced in Kenya about 36 years ago in 1984. This was under a a development cooperation project in the field of applied research for aquaculture development between the government of Kenya and the government of the Kingdom of Belgium. Now, by the end of that project, after 2 years, Artemia had spread to all the 7 corporate salt farms and all the other, having been introduced in the first 2 salt works, that was Kurawa and Crystalline Gongoni, which that time was called Fundisha. Now, at the end of that, the technical feasibility of Artemia production had been established, and Artemia was now abundant in all the salinas in the coastal salt works of Kenya, and it could be considered as a new resource that could be further developed, either for commercial exploitation as a life source for aquaculture development or for improved salt production. Now, at that time, our desire was that the next phase of the project was to enter into commercial exploitation, where the economic value of different Artemia products could be evaluated and demonstrated. However, things didn't work quite as planned. Next, please. So we then got lucky again about 10 years ago when we got a new project that was supported by the Belgian Inter-University Council, which was named the Own Initiative Project. This project basically the project was to build the capacity of local scientists and stakeholders for production of Artemia, and basically to get up-to-date knowledge and practical experience for aquaculture development in Kenya. And its overall development objective was to improve the living conditions of rural communities within Kenya through production and application of Artemia in local aquaculture ventures, meaning that we needed to create extra employment and generate income. Next, please. Next slide. So we had 7 partners: the Kenya Marine and Fisheries Research Institute, the Laboratory for Aquaculture and Artemia Reference Center of Ghent University. Kanto University was one of our project partners, and we had 3 corporate salt farms and 1 artisanal salt farm. Next, please. Now, this Google map shows the project site. You can see here there is this is Munarani Salt Farm, is the artisanal farm with whom we worked, and it is just adjacent to the biggest salt farm, that is Kent Salt, with about with over 5,000 hectares of salt farm. So we worked with both these two salt companies. Our experimental sites, however, experimental ponds were within Munarani Salt Farm. Next, please. Now, from the onset of the project, being that our main aim was to build capacity of the local stakeholders, capacity building started right from pond construction. So we involved the community in building the ponds. By the end of it, they were really experts in pond construction and everything that was to do with the project. And there was a very positive socioeconomic impact, because these people could get employment. The youth that were causing trouble got out of their antisocial behavior. The women could build houses, buy livestock, and do all sorts of other things. And having built their capacity, we were able to employ some of the local community members actually to manage the the ponds for us, the experimental ponds after this. Next slide, please. So that that picture illustrates the layout of the experimental ponds. You can see here we have 2 fertilization ponds. There is a reservoir pond, and then we had 18 ponds, experimental ponds, and 2 crystallization ponds. Next slide, please. So pond management had to be robust in order to get good production. So we had fertilization, as I indicated, and we raked the ponds and generally did very, very robust management of the farm. Next slide, please. Again, you can see there is inoculation of the ponds up there, and then monitoring of the water parameters, the salinity, the temperature, and all this had to be kept under control in order to get a good production. Next slide, please. So one of the things that was done was to train different groups of stakeholders, including the community members, the salt companies, different levels of students from school and college students and university students were trained. And we did several field demonstrations in Artemia corn production and field processing of the cysts and even quality control. Next slide, please. So that's one of the workshops that we did. This was done within the laboratories. So here we did training for production quality control and application of Artemia in aquaculture. And it also involved both the local community members from the Kenya Marine and Fisheries Research Institute and other public institutions like the Fisheries Department and the university and other college students. Next, please. And so we brought on board the top level policymakers to create awareness and also to disseminate. During this event, we had an official launch of the project where we got the minister and top levels government officials from the ministry to participate in this. And it really gave us a lot of publicity and spread the news about Artemia and aquaculture. And it also helped the community to resolve some of their issues, including land tenure system, because they could reach the minister at this point. Next, please. So at the end of our project, there was a possibility to upscale what started with the project when there was a World Bank project called the Kenya Coastal Development Project, which supported these artisanal farmers to construct 18 more ponds. And they got technical support also from Kemfree to train them and more or less optimize the production. During the end of this season, we further research indicated that the Kenyan cysts were very high in EPA and also had the same similar size to the parent Artemia of the San Francisco Bay. Now, what is even more significant is that within one season of production of 6 months, in 1 hectare, it was possible to produce 38 kilos of dry cysts and 250 kilograms of biomass, which is valued at around 5,000 USD. Next, please. So the last interventions have given birth to another project, which has just started. This project is now supported by the Western Indian Ocean Marine Science Association, and it is also aiming to optimize Artemia production technology using biofloc technology. So it also has partners from Maseno University in Kenya, the Kenya Marine Fisheries and Research Institute, Tanzania Fisheries Research Institute, Ghent University is part of this, and Gothenburg University. So we also have 2 salt farms, one artisanal and one corporate in Kenya, and 2 other salt farms in Tanzania working in this. So the project aims to characterize the local Artemia biotopes in Kenyan and Tanzanian coastal saltworks using molecular biology and determine the genetic adaptation of Artemia biotopes. They also would like to apply biofloc technology for mass production of Artemia, biomass, and cysts, and then to promote the utilization of Artemia cysts and biomass in emerging local aquaculture activities. Next, please. So finally, what we see as key milestones from these initiatives is really the adaptation of Artemia use of Artemia in local interventions. This picture shows some of the trainings we had for the local hatchery managers. So we can now we've worked with a cross-section of them, and the hatchery managers can now use Artemia cysts and biomass produced in Kenya, which the salt farmers sell to them. And though it is still in small quantities, but there is a great opportunity to upscale this. Again, we have managed to reinoculate Artemia in most of the salt works, and these salt works now do routine biological management of their salt works, and we've also established working relations with them. The other thing is, like, the largest salt farm has introduced this practice actually in Tanzania, where in Tanga area of Tanzania, where they have solar salt works, and this which is Ken salt, they have interest in that salt work. So Tanzania has now Artemia, and that is part of the reason why we have this collaboration in the last project. We also managed to recruit 25 artisanal farmers in Artemia pond production and trained 3 technicians, 2 scientists in Kanto University, one of them having been from one of the local communities in both Artemia pond production and laviculture. We also, under the project, we also trained 2 MSCs in Ghent University. And finally, Kemfree has managed to procure the first cyst processing equipment in Kenya. And now we have employed 2 technicians that work with the salt farmers. And so we have built some capacity, and there's a good prospect to upscale Artemia pond production and develop aquaculture in Kenya in the final analysis. Thank you very much.
I'm Wei Jun, working in the Zhejiang University College of Life Sciences. Actually, I'm a new face in the research of the Artemia. Yes, about 10 years ago, I met the Patrick and James Clegg, and I started Artemia. Before that, I'm working on the molecular cell biology of the cancer. Today I just talk a small talk about my work, regulation of the cancer cell dormancy using the diapause genome of the Artemia. Yes, next slide, please. As you may already know that current tumor therapy often targets the primary bark, tumor bark, but fail to eradicate the some therapy-resistant dormant cancer stem cell. This stem cell is very dangerous because they can become activated to initiate recurrence and or metastasis beyond the therapy. So about 10 years ago, I started my work, but I don't have any idea to get the dominant cell mechanisms. So this is one. Next slide, please, Patrick. This is one case of the breast cancer. The difficulty in the eradicating these dominant cancer stem cells remains as the major challenge toward recurrence-free remission beyond clinical treatment. Sometimes this dominant cell can keep the dominant status in the human body for even 20 years, and then they can wake up and make the tumor recurrence and metastasis. So next slide, please. But up to date, however, the determinant of the cellular dormancy and how they sustain themselves to generate tumor and relapse beyond the resistant to chemoradiotherapy remains unclear. Nobody to know about the mechanisms of the dormancy. So the one idea starts in my lab from the Artemia before 10 years ago. A lot of the idea is after I talked with James, Clegg, and Patrick, and then I start my work in my lab using the Artemia as a study model for the cellular dormancy. Next slide, please, Patrick. So about past 10 years, we screened a lot of the protein molecule to hope to find out the determinant of the dormancy. One result showed here. One we call that is SETD4, set domain containing protein 4. This is one protein from the Artemia. We to see this this picture in the control, the SETD4. If the Artemia expressing this protein, the Artemia keep the diapause long time. But in my lab, we used the RNAi to knock down this gene, and then found this Artemia can weaken up and then to develop to the nucleus. So we start this protein to from the Artemia to cancer. And then next slide, please. So we found that the evolutionary conserved mechanism of cellular dormancy regulated by this SETD4 in Artemia, also in cancer. This is in this case use the breast cancer cell. So absolutely the same mechanism we can see in the Artemia, also in cancer. So we start using the Artemia. That means this the research method is make our very clear to.
Wei Jun, your sound is off. Please, something wrong with your microphone. Wei Jun. Hello. Wei Jun, something wrong with your microphone. Maybe I suggest that we stop here the presentation of Wei Jun, and hopefully he can restore the microphone. And in the meantime, we move on to the last presentation. Wei Jun is back?
Yeah.
Okay. We are on the slide. We found that an evolutionary conserved mechanism. So you can.
Oh, yes. Okay. Sorry about the connection. It's not very good here. So so we found that this mechanism is evolutionary conserved because it's from the Artemia to mouse, even to human. So that means we can use the Artemia, use this model can give the more application in human cancer. Next, please. Patrick, next, please. Okay, this is the last slide. Now we found a couple factors to regulate the dominant tumor cells. So one strategy is keep the sleeping. We call that a sleeping strategy to maintain dominant cancer stem cell in harmless dominant state. To keep this stem cell in human body long time, even the lifetime. So this is the first choice. The second choice is the waking up and destroy or eradicating the dominant cancer stem cell for recurrence-free recovery. So in this case, we use the breast cancer. We successful to make the breast cancer kill all of the dominant cancer stem cell, and then totally to remove the tumor from the mice. So now this is just a small step using the Artemia to get the important gene or factor, and then to push our study in the cancer research. This is my talk today. If you have the question, I want to answer to you. Saxon.
Well, I have received just half an hour before this session started a text and the PowerPoint presentation of Ludmila. I will make her text available to everyone because it is a long but very good text. I will just read a few sentences and then go through the presentation. These are figures on the expectations of the increase in production, so what they were harvesting and where they are making some predictions on what could be harvested in the future. This is the site in the Altai of the lakes that are yielding the biggest quantities of products, as you see here, but also the ones that have been studied in a lot of detail. And again, the information can be found in the papers that were published by Ludmila and her team. So this is where she is indicating the occurrence of the different strains. I was referring to Urmiana and Salina here in the Krim, some bisexual species that might not have been described in detail yet in Russia and in Kazakhstan, and then also occurrence in the southern parts of Russia of Artemia sinica. I will not go into the details here, it can be consulted later, but you have a lot of data here on the characteristics of the Artemia sites. They are also characterized, a lot of these sites, by shallow waters, which means that there will be fast evaporation and a fast increase of the salinity in the summertime. Very different chemical composition, from dominant in sodium chloride, others sulfate, even carbonate. And of course, we have the difference here between the summer and the winter, where in the winter time, the cysts are surviving. You have hatching in the springtime, nuclei and juvenile production. They grow out into adults, and you will have an overlapping of different age groups. And probably when winter approaches, in autumn, maybe some more stressful conditions, and that's when cysts appear. Some data here on what amounts of Artemia product is harvested from the different regions. And remember from one of the previous slides, it is in the Altai region that about 65% of the commercial harvests are made. Interesting also to see about who is importing these Artemia from Russia and Kazakhstan from the latest statistics. China, Thailand, Germany, Turkey, and a number of other countries. Just again, some data from another publication, what is coming out of the Great Salt Lake, and estimated what comes out of Russia, Kazakhstan, Uzbekistan combined, and China. I have not had the time to look into this in detail. So harvest in Russia, metric tons brined product over the number of years, and the correlation with a dry year, a wet year, and an average year. Same here when they express it in kilograms per hectare. Data on the phytoplankton and zooplankton composition. This is, of course, for the salt lakes where they start from a salinity that might be in the order of 40 to 50 grams per liter up to over 100 grams per liter. So when you see, excuse me, presence of rotifers, copepods, and other crustaceans, this is the salinities where you do not have Artemia. So this is the euryhaline species, but up to a limit where might be in the order of 80 or more grams per liter, whereas at the highest salinities, where you call then the halobions, you have Artemia and the brine fly Ephedra. The evolution in function of salinity, again, we have to look at it in more detail with more time. Characteristics of the morphometry of the Siberian cysts. You see that there is a lot of variation, with some real big ones in diameter of the cysts, and then some others that come closer to what we see in the Artemia franciscana species. Ratio between salinity, Artemia weight, number of brood per female. So you see that there is a lot of scientific information available, published by Ludmila and her group, and can be consulted if you want to have more details. Main factors for generation of medical mud on Meveji Lake. So thalassotherapy is practiced in quite a number of these salt lakes. And it's interesting also to see the, well, the classic ecological calculation that is made, how much phytoplankton is needed for the production of 100 kilogram of cysts. Because the recommendations for allocating permits and quota, these are only 1/4 from the total stock. Therefore, it is considered to be that enough cysts are remaining for future repopulation. Some forecasts here for the period 2015-2016. The details that you have seen earlier, but with more detailed figures for the export from Russia and Kazakhstan to the countries that I have listed before. And finally, some general pictures on the monitoring of the Artemia cysts in Russia. Most of the harvesting is done from the shoreline. Bolsho Yarovoy apparently is also an area where there is harvesting in the water. Also, the equipment that is used to go on the lake to take samples of the Artemia biomass and do analysis on the density of the cysts. You see some of the conditions under which you were working in these Salt Lake shores, and here from the boat, the plankton samples and the analysis of the plankton samples.
Good afternoon, friends and colleagues. Saxon and Patrick, congratulations for organizing this session on Artemia, and thank you for the kind invitation to participate. I am Eddy Naessens from Aquaculture, which is the advanced nutrition business area of the aquaculture technology company Benchmark. My involvement with Artemia started in 1984 with projects first in Kenya, later in Ecuador. Then followed a period where my main activities were related to Artemia sourcing and production, and the product development at Aquaculture. Currently, I am responsible for the Artemia market intelligence within the company. In practice, this includes the follow-up and mapping of the global Artemia situation. This in regards to availability, supply, and the general application of the resource. In this present presentation, I want to give you a brief historical overview of some Artemia aspects in which Aquaculture has been involved. More specifically on the sourcing and the upgrading of the use of Artemia in global aquaculture. Everything started in the University of Ghent within the laboratory of Artemia Reference Center. It was at 70s that the FAO endorsed ARC as the World Reference Center for Artemia research. The work at the ARC focused mainly on fundamental research, but also on practical on-site applications, such as the development of the controlled production of Artemia cysts and biomass. In 1983, out of this Artemia Reference Center, a spin-off company arose with the name of Artemia Systems. The purpose of this company was to translate the findings of the Artemia Reference Center into commercial relevant applications. Later on, in 1991, Artemia Systems was integrated in the Group as Aquaculture. Aquaculture was and is specialized in the nutrition of larval species of marine fish and shrimp. Part of that dealt, of course, with Artemia as the crucial component in the larval nutrition. At these moments, the company engaged in the production of Artemia cysts, such as on the Great Salt Lake in Utah, as well as the optimization of Artemia use in aquaculture. Later on, in 2016, Aquaculture was embedded in the diversified aquaculture technology company Benchmark, where it continues and intensifies the developments for facilitating and rationalizing the use of Artemia in the growing global aquaculture markets. From this overview, you can see that the Artemia activities coincided with the rapid expansion of commercial aquaculture in the 80s and 90s, and in the subsequent exponential growth of this commercial aquaculture. But along the way, it was seen that Artemia became more and more critical. And this was shown in a variety of problems, such as the supply of Artemia. Will there be sufficient Artemia to support the anticipated aquaculture expansion? Another problem or issue is, will this Artemia that is provided to these markets, will the quality of this Artemia be sufficient or good enough to feed the growing number of cultured species? And finally, there was also it was also seen that the use of Artemia in aquaculture was far from optimal, and a lot of Artemia was actually wasted. A lot of this value of scarce resource was actually wasted by inappropriate use. So it was clear that in order to keep Artemia as the driving force for aquaculture, in order to keep this Artemia there was action needed. And within the company, within INVI Aquaculture, there was the Artemia task force was created, which had 3 objectives. The 3 objectives that were actually there to tackle the 3 main problems here. The supply of Artemia, the quality of Artemia, and the rational Artemia application protocols. INVI had the intention to implement, to obtain these 3 objectives by means of use of its in-house expertise, but also by collaboration with specialized partners, which are which can be could be universities, laboratories, but also local commercial enterprises that were dealing in Artemia. So the approach was really a multidisciplinary approach. Looking at the first objective, the diversification and the stabilization of global Artemia supply, the tasks to obtain these objectives were defined as site service and explorations, which would be needed to do ecological studies to evaluate the economical relevance of newly defined sites of exploitation. And once this was done, of course, there was also the real valorization itself, in which INVI engaged in the establishment of local representations, the training of local staff, the installation of infrastructure for harvesting and quality control, and then finally the commercialization itself, being locally or for export. The first site, the first area, region that was tackled in this exploration was Russia. And within Russia, 2 main areas, the Altai area and the Kurgan area. In both areas, the INVI established local representations, local companies, which were called INVI Altai and INVI Kurgan. Next came Kazakhstan and Uzbekistan, 2 other countries of the CIS. And in Kazakhstan and Uzbekistan, in Kazakhstan, we had 2 areas, actually 3. First, there was Western Kazakhstan, where there were a number of lagoons located near the Caspian Sea. There was East Kazakhstan, close to the Russian border, the Pavlodar area. And there was also the area around the Aral Sea. The same Aral Sea had also parts in Uzbekistan, and also that Uzbek area of the Aral Sea were explored through this Artemia task force. At that moment, in the local INVI representation was in Kazakhstan, close to Aktau, the city of Aktau, near the Caspian Sea. A third place in the CIS was Turkmenistan, where we concentrated on the northern, very big lagoon, the Karabogaz Gol. And we had a representation in the capital in Ashgabat. Another huge area was China, where we had the company TIAC that was coordinating the explorations and the valorizations of the local Artemia biotopes, which were situated along the Bohai Bay, in Inner Mongolia, in the Tibet area, and also in the northwestern parts with big lakes like Ibi and Balikun. So after all this work was done, and when we look back, we started with Artemia, commercial Artemia production only in the Great Salt Lake in Utah. After the exploration through the Artemia Task Force, we had new areas, new regions from which Artemia could be harvested in the CIS and in China. So if we look at the productions before, or the productions from the Great Salt Lake here in the blue bars, after the explorations of the Artemia Task Force, these blue bars were actually extended with the red and yellow bars from CIS and China. And we really could say that we fulfilled the first objective. It is increasing and stabilizing the Artemia supply worldwide. We could say that after that, the global, the total Artemia production was around 2,500, 3,000 tons, which was rather stable and which was sufficient for the aquaculture needs. Apart from the supply, there is the second objective, which was the Artemia quality. There we, together with the partners in the Artemia Reference Center, we developed the standards for hatching, standards for, we also characterized the Artemia. You can imagine that if we drew Artemia from so much different origins, Great Salt Lake, CIS countries, China, we could expect that this Artemia was not all the same. So a main objective in the Artemia Task Force was strain characterization according to, for example, biometrics, and also according to the nutritional value. In the second objective, the implementation of Artemia quality, we also engaged in the product upgrading. We were aiming at making the Artemia that were delivered by Artemia, but by nature, sorry, by making these Artemia more user-friendly. And two examples here are the development of Artemia that had separation characteristics, which were actually supranatural. The separation, the complete separation of naupli from the cyst shells was done via technology in which magnetic forces were applied. So because of this upgrading, we could make Artemia more user-friendly. Another upgrading of the Artemia was not so much user-friendly, but making the Artemia more friendly for the predator, for the fish and the shrimp larvae. It was possible to create, to modify via processing techniques, to modify the Artemia into an animal that was free of bacteria, which is not the case when you use just natural Artemia. Supply we had, quality we had, now we have to use this sufficient quantity of Artemia of good quality. We have to use it optimally in the hatcheries. We have to develop adequate Artemia application protocols in order that this critical and scarce resource was not wasted. And in that respect, instructions for use were developed, instructions for the optimal the optimal yield of Artemia nobili from a given quantity of cysts, the storage, the maintaining the good qualities over a longer period. These were actually the application protocols, the instructions for use, which were defined and that were taken over actually by the whole industry. If now you look at a label on a can of Artemia, you look at the instructions for use, it's these instructions, it's the instructions for use that were established by the Artemia task force. So we talked about the supply, the quality, and the application. And I think the objectives of the Artemia task force by this were fulfilled in order for continuous support of the aquaculture industry. Thank you.
Now, obviously, all of you have covered, very kindly covered many different areas, speaking regionally about the history of Artemia. I have a number of maybe specific questions that perhaps, if you don't mind, I'll follow up with you individually by email. But just to ask maybe a more global question, perhaps each of you can take maybe 2 or 3 minutes to answer 2 questions from myself, which is, moving forward, you know, a lot of the presentations were about history and what is. Looking to the future in each of your regions, if you could tell me what is the biggest concern or challenge for you at the moment, and part 2, what would you say is the most exciting area of potential moving forward?
Yes, I only got the first question. So I think the big challenge for Artemia cyst production is the unstable quantity of cyst production. You know that the most of Artemia cysts are obtained from the salt lakes and the dry out, or sometimes the dry out of the salt lake. The high salinity or the very low salinity. All this will influence the production of the Artemia cysts. I think this is the one of the challenge. Saxon.
Thank you so much for that, Liying.
Yeah.
I will offer the second question to you for you to answer, but given your specific role in the Asian Artemia Reference Center, just a side question to yourself, how are you working to impose regulations in terms of harvest management systems? In my research, that's obviously been something crucial for GSL and how they've emerged and how they've been able to sustain. Obviously, it fluctuates year on year, but how they've been able to sustain their Artemia production. So how are you helping different productions in Asia with this sort of harvest management system? And the second question was, what is the most exciting area of potential for your region in terms of the Artemia industry? Thank you very much.
Yeah, okay. So yeah, as we all know that the Great Salt Lake, the cyst harvest from the GSL is very well organized for many years. Unfortunately, I don't see the similar management or organization have been done here in China and also our neighbor countries. I think it's also one of the tasks of ARC in the future to try to give advice, or of course on base of the survey on how to harvest the Artemia cysts in a sustainable way. But I think so far there's no one lake can do it.
Thank you so much for that. And if you wouldn't mind answering the second question, for you personally, what do you think offers the most exciting area of potential in the field of Artemia in
yes.
in Asia?
Yeah. I think for me, because some of my colleagues and even the director of our university always ask me, for your center, can you develop Artemia into a food, into a human food? You know, as I think Hoa has mentioned that in Vietnam and also in another Asian country, they do cook Artemia, the omelette, as a human food, but in China, not yet. So I think it's, I think the most exciting point in the future in our center that we try to also to develop like a protocol or to study on the how to make Artemia as a human food, because it contains a lot of the proteins and also the lipids and especially the fatty acid.
So for the first questions, I think for Thailand, you can still hear me? Okay. For Thailand, although we start to produce this, but later on, the biomass is the requirement for the farmer. And also the Artemia can adapt very good to the cultural conditions in Thailand. So although we try to produce this, but in the opposite, the Artemia, they produce more on biomass with less seeds. And nowadays, the demand for Artemia biomass is decreased because Thailand has a higher activity of Pinus vanamai, which is now account for more than 90% of shrimp production, while the Pinus modern is gets decreased to only 1%. So it means that the demand for Artemia biomass is also decreased because Pinus modern, they are more cannibalistic, while Pinus vanamai, they are more omnivore, so they require less of Artemia. So that's why we get low demand of Artemia biomass. And also, I think the price of Artemia biomass is getting down. The farmer, and also the hatchery, they use the artificial diet more than Artemia biomass. So I think if we can culture or have the new species like a crab or swimming crab or mud crab that they maybe require more Artemia biomass, it can again increase the activity of Artemia culture.
Indeed, aquaculture activity is growing very fast in Iran since last 2 decades. And recently also very big investment for the production and culture of marine fish, which also requires lots of Artemia. And shrimp farming is also expanding very fast. So what we need is lots of Artemia cyst. Currently, the consumption is not so high, maybe about 40 tons of dry cysts per year, but Urmia Lake has no production, therefore we have to produce it artificially or we have to import. So the production in artificial ponds are not so much currently, so we have to import a lot, but with increasing activities for the shrimp production and marine fish and also sturgeon fish culture, and also for the aquarium fish, we need lots of Artemia. So I think we are obliged to go more and more on the production of artificial production of Artemia in different areas, especially where we have lots of different types of salt water, as I mentioned in my presentation. We have Persian Gulf and Oman Sea in the south of Iran, which are having salinity of about 40 grams per liter, but that area is very hot, so we can increase the salinity by evaporation. This year, especially the hatcheries, especially the shrimp hatcheries, had lots of problem to meet the Artemia cyst they wanted, because the production is being increasing very much, and the Iranian currency, unfortunately, it has lost its value so much during the last 2 years by about maybe 4 or 5 times. And therefore, it is becoming so costly for the Iranian farmers to import the cyst from abroad. Therefore, it is a very big challenge for us to make a very uniform protocol for production of Artemia in different areas, and also use more and more biofloc for production of Artemia, and to increase the production artificially in the country. And we have, of course, we have lots of different kinds of saline salt waters which can be used for this purpose. And the potentials, as if I have to mention, so may I go to the next question, or it is not enough with the first one?
Yeah, please go on to the exciting opportunities. Thank you.
Yeah. So potentials in Iran, of course, we have very good potentials for the aquaculture activities in Iran, for both for marine and also for the freshwater aquaculture. We have lots of very different kinds of water resources in the country. In north of Iran, we have the Caspian Sea, which is very good for the sturgeon fish. As you know, the Caspian Sea was the biggest biotope for the sturgeon fish before, but now, unfortunately, we are losing that. So therefore, in 25 provinces of Iran, sturgeon fish aquaculture has been started, and they are producing artificially caviar also. So also they also need, these are the potentials. And in south of Iran, we have the Persian Gulf and Oman Sea for shrimp and marine fish production. So we have lots of potentials to improve the aquaculture activities, but however, most of them need Artemia as a food source for the early larvae. And also, of course, we have been working, some of my colleagues have been working on production of biological active compounds, especially different carotenoids from Artemia and also from other crustaceans. And also they make different types of cosmetics which could have, which can catch much more price compared to Artemia.
Yes, thank you so much for that, Naser. And in terms of your presentation, one thing that I found very interesting, let's say, is using effluent from urban treatment plants as a supply of water and food. I haven't heard of that before, so very, very interesting. I think now we can move on to Betty. So if I could ask you the same 2 questions, that'd be brilliant.
Yeah, so thank you very much. Concerning our biggest concern or challenge, probably I could say, like I mentioned, Artemia was introduced in Kenya about 36 years ago. Now, I think one of the biggest challenges for us as to why we didn't move forward as much as we would have wanted is the fact that there was no support at the policy level. So for a long time in Kenya, support for the fisheries sector or aquaculture per se has not been forthcoming. It's just in recent years that we're starting to see a change in policy. So that has been a big challenge. So when there's no recognition at the policy level, and then you don't even get the kind of support you need to promote the sector in terms of capacity building or research. And so that has been a big challenge. And the other thing is, much as we had this Artemia right now in the salt works, and we have the 10,000 hectares of salt works, but its promotion to a commercial level was not forthcoming because the majority of salt farms are these big corporate salt farms who are not really interested in producing Artemia as a commercial product, because for them, they make so much money from salt, and doing something like Artemia production for them looks like, you know, a sideshow. They would rather use it for biological production, and they think it's rather a waste of their time. So the idea is, how do we get to convince these people that they can produce this, and not only to make a little more money, but also to support the aquaculture sector. And it's easier to work with the artisanal farmers for production of Artemia. But then the challenge we have in our land tenure system is that the small community members don't have access to land. So that's a very big challenge for us. They are willing to work, but they don't have long-term, you know, access to the land they have. Then we also have currently uncertain weather patterns. Sometimes you get, you know, extended rainfalls, and so that poses some level of a challenge. Now, concerning the exciting areas, for one, there is a high demand for Artemia. There is significant demand because there's a lot of promotion of sustainable food from the sea right now. That will continue, you know, increasing the demand for Artemia, particularly in the sustenance of a sustainable ocean economy in terms of combating climate change and so forth. There is a push to produce more sustainable food from the ocean. And also for us, our cysts were very popular with our local farmers. They preferred our locally produced cysts because they found that they had better performance than you know, the imported cysts. So there is great motivation. We have an opportunity and a prospect to develop that sector. And finally, we have for the first time we're getting a mariculture center being built. So we have opportunity to develop, you know, the different aspects of mariculture. So thank you.
Yeah, thank you, Saxon. From an aquaculture point of view, it is clear, and it was clear also from some of the presentations, that Artemia is and will be an important item in the aquaculture. I think I have heard and I've seen on some presentations some words like illegal harvest and poaching and this and that. I think since Artemia remains very important, we must be very careful that it remains sustainable and that there is no over-harvesting, illegal harvesting, poaching. I mean, that there is good regulation with the Great Salt Lake undoubtedly as the example, good regulation about the harvest quota and the harvest regulations. I think this is crucial for the sustainability of the Artemia as a resource, indispensable resource for aquaculture. I think that is a concern.
Thank you so much, Eddy. And would you like to take a minute or two to talk about what you find sort of most exciting looking forward?
Well, most exciting is for me the opportunity that Artemia offers as a model organism in so many other fields that are unfamiliar to me. I'm thinking about the cancer research and the opportunities that are looming at the horizon there with Artemia. I'm also thinking about a presentation of yesterday, a presentation of Jim Clegg, where he said that taking advantage of the unique properties of Artemia, researchers have been able to study and really develop something what he called like dry blood. There is a high need of blood for medical reasons, but blood is a very bulky substance. And if, because of the making use of the unique opportunities of Artemia, you can turn this bulky quantity into a much lower, much more transportable, preservable unit in the form of dry blood. This for me was also something exciting to hear.
Yeah, most definitely. What a smooth transition that is as well to the segment of Wei Jun. So thank you for that, Eddy. Wei Jun, I wanted to leave you for last because it's obviously slightly different in terms of the perspective that you're going to offer. And obviously all of what you're studying is very, very exciting and crucially important. So perhaps I should rephrase the questions. I would still be curious if you do have challenges and concerns that are barriers in terms of your research, but perhaps you can speak more on what your priorities and focuses are in the next 5 to 10 years with your line of research. That would be really fascinating. Thank you so much.
Okay. Actually, I will to explain why we can use the Artemia as a study model, because we can harvest a lot of the Artemia at the same diapause stage. But in the cancer, we just in one cancer, just a very small population of dominant cancer cells. That's why we cannot use the cancer to study cancer. We can use the Artemia to study cancer. That is much good way to focus on the specific focused on the dominant cancer cells. So in the future, I have the plan to try use the Artemia as a model to find more factors that will regulate the cancer's dominancy. So I hope, because we just focused on the evolutionary conserved mechanisms, that is that means the same thing occurred in Artemia and in cancer. So now this is a big challenge to use to find out the evolutionary conserved mechanisms. In my study, we just found 2 factors that's conserved, but many of the factors is specific for Artemia, but not the same in the cancer. So that is a big challenge. Another big challenge is up to date, we still don't have the genome database can be used in the Artemia study. So that is I hope in the Patrick group can publish faster about the Artemia genome data. That is it.
Thank you so much for that, Wei Jun, and a good summary for the event. Patrick needs to work faster.
Yes.
We'll pass back over to you, Patrick, and perhaps you can wrap up this incredible talk. But from me, thank you so much, everyone, for being involved. You've given me an incredible amount of homework, which I probably will never be able to complete, but I'm so grateful to be involved in this talk. Thank you so much.
Yeah, maybe Saxon, in the meantime, Hoa has answered in the chat box. So, and indeed, the big challenge is the weather change. This is a general concern that I like to add. Climate change has a lot of impact on a lot of these salt works and places where Artemia is being harvested. Then the second challenge they have is Artemia areas have been switched to shrimp farming as they make better profit. And for opportunity, he is mentioning high quality cysts produced in the site and will be expanded into the areas nearby. Vietnam has a demand of over 500 tons per year for aquaculture. Today, the production only covers 3 to 5%. And then more and more biomass have been interested, not only for aquaculture, but also for other purposes like food. If you allow me, Saxon, I would just like to comment on the different interventions that we have just heard and summarize a bit. And this is, in fact, the main reason why FAO wants to promote the setting up of that new International Artemia Consortium. First, as we heard from Eddy and from others, to consolidate the use of Artemia in hatchery aquaculture. This is really crucial for the future. And I think with being for more than 90% today dependent on wild sources, what we have heard from several speakers, Hoa, Pam, Naser, and Betty, opportunities to produce Artemia locally. And produce Artemia locally also with a socioeconomic dimension. And I'm referring to the many, many areas in Southeast Asia, and I'm sure also in Africa, where you have the artisanal salt ponds, where you have the poorest of the poor people living, and where you can bring in a new integrated activity. So one. Second is that there are many areas in the world. Naser was giving an example. Last week we were discussing about Uzbekistan. There are many countries in Central Asia where they have salt-affected soils that are good land for agriculture, but that cannot be used anymore for classic agriculture. These are areas that could be converted into new food production activity. When I say food production activity, I'm thinking about, of course, product for aquaculture, but even also product as an ingredient in human foods. What I enjoyed hearing from several of the presentations, and that links very well with also the discussions at the United Nations level about Food 2050, we need to go for more integrated farming. And the example that Naser was giving, the example that Pam was giving, using effluents from aquaculture farms. There are good examples in Thailand. There is a very nice model system. I know that Hoa is also working on it, where the effluents of the shrimp ponds containing nutrients can be in fact put up into a higher value by evaporation and Artemia production. Use of the effluents from desalinization, as some of you have mentioned. Agricultural byproducts that are considered now as waste that could be converted. So this way, I think there are new socioeconomic opportunities, and at the same time, it will make a better balance that, okay, we need the bulk of the Artemia for aquaculture in the future from the salt lakes, where hopefully there can be more sustainable harvesting and hopefully not too much negative impact from climatic change, but that this way also there is a balance in provision. And that finally meets very well the SDGs of the United Nations, so Artemia can really play a nice role. And I'm referring to what I still need to send to some of you, the paper that FAO has just published 1 or 2 weeks ago on the role that Artemia can play in fulfilling some of the SDGs for the future. Thank you. Back to you, Saxon.
Thank you so much for that beautiful conclusion. Yeah, I sort of said my part already. Again, I am so grateful for all of you to take so much time out of your day to attend this and to share with me your experiences and knowledge. I'm extremely grateful. If you don't mind, I probably will be in touch at some point by email, so you haven't seen the last of me yet. And yeah, I really hope that this consortium that Patrick is working on can, in the future, you know, sort of better unite you all and unite the rest of the world in terms of how Artemia is going to play a role in many different areas of our lives. So thank you so much. Unless you have anything else to add, Patrick, I think that was really fantastic. Thank you, everyone.
Okay, everyone, have a good day. Thank you.